Hi, I'm Emilie G, and I'm here to talk to you about the reason you are functioning. Cells. They are often overlooked, but are the basic building blocks of all organisms. I'll begin by explaining the history of cells and how humans discovered them, and the development of the cell theory, which is basically the constitution of all cells. I will also transition into talking about morphology, aka the shape, structure, form, and size of cells. In microbiology, cell morphology is essential to understand to be able to identify certain bacteria. Now let's get started. The discovery of cells. Cells were discovered in the 17th century by a man named Robert Hooke. Hooke discovered cells through a honeycomb-like structure in the slice of a cork. He noticed compartments and called them cells. This piece of cork he observed was a dead piece of tissue, so he was only able to see the cell walls. Hooke's observation inspired many other scientists to study cells, which later became known as cell biology. It wasn't until years later when living cells were seen by a Dutch biologist named Anton van Leeuwenhoek. He made a special lens in order to observe a pond in hopes to find some evidence of living cells. 169 years later, after multiple scientists continued to view cells and study them, the cell theory was finally accepted in 1839. In 1839, Theodor Schwann concluded that cells make up the tissues of plants and animals. Although both plants and animals are made up of cells, the cells were different in structure. He came up with the three basic principles of the cell theory. 1. All living organisms are composed of one or more cells. 2. Cells are the basic unit of structure and organization in organisms. And 3. Cells arise from pre-existing cells. Schwann was not alone in influencing others to accept the cell theory. He had help from other scientists named Schleiden, Remak, and Virchow. The cell theory was later modernized and added three more hypotheses. 1. Energy flows within cells. 2. Genetic information is passed from one cell to the other. And 3. The chemical composition of all the cells is the same. All this talk about the cell theory, and we haven't even discussed cells themselves. So, what exactly are cells? According to the cell theory, cells are the basic building blocks of all living organisms. They are the smallest independent component of an organism, and they can replicate independently. Although there are multiple types of cells, they all have two things in common: a nucleus region and membrane-bound organelles. The two main types of cells are prokaryotes and eukaryotes. Prokaryotic cells are simpler than eukaryotic cells. They have no nucleus, only a nucleoid. A nucleoid is a region within the prokaryotic cell that contains the genetic material. They are single-celled microorganisms. Some examples are archaea, bacteria, and cyanobacteria. They contain DNA or RNA and are reproduced by binary fission. Now on to eukaryotic cells. Eukaryotic cells do contain a true nucleus. They are significantly larger than the prokaryotic cells. Eukaryotes contain a plasma membrane which controls the transportation of things in and out of the cell. The plasma membrane is also the way that cells communicate with each other. Eukaryotic cells need this because they are mostly found in multicellular organisms such as plants, fungi, protozoans, and animals. They reproduce sexually and asexually, aka mitosis and meiosis. Although eukaryotes are found in both plants and animals, they are not the same within each. In a plant cell, a eukaryote has a cell wall, contains chloroplasts, and vacuoles. The animal cell does not contain any of these extra things because they are because these are specific to plants only. Microbiology takes a closer look at prokaryotes and certain characteristics that prokaryotes have that are important when studying bacteria. Some of the prokaryotic cells form a layer of carbohydrates or sugars/saccharides, which are identified as the glycocalyx. This layer protects the cells from hosts and infections. It can be several different forms depending on how condensed the glycocalyx is to the cell wall. If it is very tight, it can be referred to as a capsule, but if it is very loose, it will be referred to as a slime layer. There are two main functions of the glycocalyx. 1. It causes a bacterium within the glycocalyx to become invisible to a host's immune system. This invisibility makes it harder for the phagocytes to surround and engulf it. Infectious strains of bacteria like Staphylococcus, Streptococcus, and Pseudomonas all rely heavily on the glycocalyx compared to non-infectious strains. The second main function of the glycocalyx is a protective layer that is formed to cover and protect the cell, which makes it hard to kill. This layer is called the biofilm. Some examples of a biofilm are plaque on teeth or pond scum. Both of these are essentially buildups of bacterium. Most eukaryotes are present in larger organisms, but sometimes they are in microorganisms. The eukaryote microorganisms are fungi, protozoa, and simple algae. Flagella and cilia are two important components of eukaryotic cells. Although prokaryotes have a flagella as well, they have a different purpose in eukaryotic cells. The eukaryotic flagella is much more complex than the prokaryotes and functions as a way for the cell to move by causing a whipping movement. The cilia cause a back and forth beating around the cell. It can function as either a form of protection but also assist with transportation. Cell morphology. Cell morphology is basically just the identifying of the shape, structure, form, and size of cells while they are being studied under a microscope. It is important to identify the morphology of the cells because it can sometimes indicate a contamination, infection, or health problem. There are three main categories of morphology: cocci, bacilli, and spirilla. Cocci is more of a circular shape, while bacilli is more like rods and spirilla is squiggly. Within each of these categories, there are arrangements. Cocci has single diplococci, which is 2, streptococci, which is a lot in a row, tetrad, and sarkenes, which is 4, and staphylococci, which is a large cluster. Bacilli has single alone, which is alone, diplobacilli, which is 2, streptobacilli, which is multiple in a row, and palisade, which is a large cluster. Spirilla only has single arrangements. On the next slide, there is an enlarged picture of the smaller one on this slide. This picture includes the different types of morphologies as well as their types of arrangements. It is from biologydiscussion.com. Each arrangement has a particular meaning behind it. When viewing a slide and you see a diplococci arrangement, it is more likely that it is indicating some kind of bacterial infection. However, this must be a gram-positive coccibacteria. If it is gram-negative, it is indicating either a Neisseria, gonorrhea, or meningitis. What you are looking at on this slide is a gram-positive diplococci arrangement of a strep throat swab. Different slides will have different colors and arrangements, but they will always have the same circular cocci shape. This slide is showing a picture of Streptobacillus mononoiformis from a patient's epidural abscess. It is apparent that it is Streptobacillus mononoiformis because of the way that the multiple rods are arranged in rows, making clusters. It is clear that this is a gram-negative result because of the pink color. This is commonly shown in patients who have rat bite fever. This is the last morphology, spirilla. Remember, spirilla is only arranged in single arrangements. The picture you are looking at is a microscopic view of the Lyme disease slide. A spirillum bacteria called Borrelia burgdorferi is one of the infectious agents of Lyme disease. One of the most significant things about spirillum morphology is that they have a polar flagella, meaning that it can rotate the cell body in the opposite direction. Conclusion. From this lesson, I hope you now understand the history of cells and cell morphology, and what cells are. In a microbiology class, these topics are essentially the cells of understanding microbiology. If you don't learn about the basics, you can't further develop your understanding. Sometimes listening to lectures like this seem like they don't apply to your everyday life, but this stuff does. Every day, you are making and losing cells in your body, while also coming into contact with thousands of bacteria that are fighting your own cells and trying to infect them. It is important to be aware of this and take the precautions to make sure to take care of your immune system so your fighter cells can continue to fight diseases. I hope that you have enjoyed this lesson and learned as much as I did this semester about cells and microbiology.